Hello everyone, so this is Group 2 final project presentation for subject Medical Entomology. My name is Farhana Athirah and I'm going to present the introduction for this final project. For this final project, we have carried out insect collection, which refers to the collection of insect and arthropods for scientific study. What are arthropods? They are the organism under the phylum Arthropods that have general characteristic of jointed legs, segmented body, bilaterally symmetrical, and under the phylum Arthropods we have 4 subphylum, which are Crustacea, Myriapoda, Hexapoda, and Chelicerata.
Hi, my name is Rinushaa. I'm here to talk about the objective of major project. So first is to identify the genus, species, the taxonomy class, the medical importance of the arthropods, and also to identify and apply the correct techniques in the insect collection and apply a correct technique for the preservation of the arthropods, and also to understand the essential characteristics of the arthropods.
My name is Razina binti Muhammad Shaharul. For the materials part, the materials that we have used to catch the insect are net, chloroform, cotton ball, moss ball, plastic containers, plastic cup, shovels, fruits and leaf, centrifuge tube, pipette, insect repellent, insect pin, plasticine and display box.
Next, I will be presenting about the method. Flying insects were captured by using a net. Soil insects or ground insects were caught by a DIY pitfall trap. Some mosquito larvae were captured by using a pipette. We killed the captured insects by using chloroform. All insects were carefully pinned in the insect collection box. All of the insects were labeled, and we also put some mothballs inside the display box so that this could prevent the fungal growth on the pinned insect.
Next, for the insect captured places, this includes housing area, MSU garden, playground area, Taman Tasik Shah Alam and Taman Wetland Shah Alam.
Next up to the insect collection. So basically we collected 12 insects total. Each of us collected 3 insects. So first Farhana got 3 insects, which are grasshopper, weaver ant and red veined darter. So she got all these insects under subphylum of Hexapoda. Next, coming up to the Jane's insect collection, she got housefly, bottlefly and beetle, which is also under subphylum of Hexapoda. Moving on next to me myself, I have also got 3 insects, which are the broaden-head sunfly, Culex mosquito and crickets. Mine is also under subphylum of Hexapoda. Lastly, Razina's insect collection, she got jumping spider, tiger mosquito and oriental latrine fly. Her insect collection goes under 1 Chelicerata and 2 Hexapoda.
Moving on with the life cycle part, here we only captured insect with holometabolous, which is known as complete metamorphosis, and hemimetabolous, which is known as incomplete metamorphosis. Insect with holometabolous includes housefly, blowfly, mosquito, weaver ant and beetle. Meanwhile, insect with hemimetabolous includes spider, cockroach, dragonfly, grasshopper and crickets.
First of all, by using a net, we swing the net quickly through the air to trap the arthropods. Secondly, we also use the pipette to suck the mosquito larva on the water surface. Lastly, we made DIY pitfall trap to trap ground insects like beetles, etc.
In the overall project, we managed to analyze our insect collection into 2 main subphylum, which are the Hexapoda and Chelicerata. Moreover, we were able to catch 1 species of Chelicerata and 11 species of Hexapoda. Unfortunately, we could not find any arthropods under subphylum Myriapoda and Crustacea, and we were not able to go to any beach site to catch. Moreover, it was really a hot summer lately, so most of the soil was dried up. Normally, Myriapoda arthropods roam around damp soil.
So in conclusion, we have a clearer picture on how to identify the arthropods and how to differentiate them from the other subphylums, and we got to observe their characteristic and their taxonomy classification. Other than that, we were able to perform the correct technique for catching, preserving and labeling the arthropods.
So for my reflection part, to be honest, I really had so much of fun catching the arthropods, as well as I have learned that we need to be more patient whenever we are catching them, because it is not an easy thing to do. And I also have learned that we need to know which types of technique that can be used to catch the insect in order to improve our catching skills.
Overall, I think that this final project is very beneficial for me as I can learn more about the common species around me, and also I learned that how important it is to have a balanced ecosystem.
So for the overall reflection, I can say that the process of completing this final project was overall fun and challenging. Since this is my first time catching insect, was definitely memorable because we gain new skills and knowledge. We know how to differentiate the arthropods and how to preserve them. Although I had some hard time finding different species at different location. Since I also lack skills since it's my first time. In conclusion, this project helped me build my teamwork skills and helped me face my fear of insects. Thank you.
So even though we faced many conflicts, the moment we started our process on catching the arthropods was really fun and became the most memorable project ever. During the process of catching arthropods, we were able to communicate within ourselves and ask doubts to each other. This shows the interest of us towards the project of medical entomology, and I personally felt happy and lucky to have such a wonderful teammates working along. And overall, in this major project, every minute things we share our knowledge. Moreover, I feel the knowledge of arthropods now is actually widened.